What we have shown in our research already is that we can actually manipulate human cells directly outside the human body and change their circuitry, alter these circuits, whether they are from inherited diseases or whether they correspond to cancer. So we know what genes to intervene in, we know what cell types to intervene in, we know what regulators to perturb, and we can actually build genomic perturbations at the DNA level or at the RNA level that alter these circuits in the exact way that we are predicting would make a difference, and we can see these differences manifest themselves at the cellular level. We have also done work in mice to basically show that we can actually manipulate these circuits in mice and show a phenotypic difference at the organism level in the mice that actually alters the disease phenotypes. I think the next hurdles are really about how do we now take what we have shown at the cellular level or at the animal level and actually translate it to humans. And that's where partnering with biotech, with pharma, to actually develop new classes of therapeutics that can target specific cell types and specific genes within those cell types, as well as new chemical entities that can actually manipulate those circuits that we have shown are crucial to the underlying mechanism of the disease, to actually manipulate these circuits in living humans. And that involves hospital partnerships, that involves clinical trials, and that involves very long-term partnerships with pharma and industry in order to actually be able to translate the findings from this academic lab to making a difference in the clinic, making a difference for patients, and making a difference in the world. So the goal of our research right now is, of course, continue our search for mechanism, continue dissecting for many different genetic traits. How do they actually work? What are these circuits? What cell types do they act in? And how do we manipulate them? Carry out these manipulations in human cells, carry out these manipulations in mice, and of course partner with industry to make these translate into better human health. Psychiatric disease has been traditionally impervious to these types of treatments. If you look at the complexity of psychiatric disorders, such as schizophrenia, for a long time, genetic approaches simply failed because there are so few individuals that carry any one of these mutations. There are so many different ways that this disease can manifest itself, so many different genes that can be impacted that ultimately lead to schizophrenia, that the previous genetic cohorts, the number of samples that we had, were quite limited. This has changed dramatically over the last couple of years. There's this very large psychiatric genetics consortium that actually gave rise to more than 100 different regions of the genome that are robustly associated with schizophrenia. Interpreting these loci is really the next challenge. So we have now started a new collaboration that allows us to take human cells from schizophrenia patients and controls, differentiate these cells into different classes of neurons and different classes of glial cells, such as astrocytes, oligodendrocytes, or microglial cells, and see how the genetic differences that predispose these individuals to schizophrenia, how these genetic differences manifest themselves in each of the different cell types. So if you look at excitatory neurons, inhibitory neurons, the genetic differences turn on different genes and they have an effect in different cell types. And understanding that is actually, again, once more, the first step to treatment. So basically what we're doing right now is studying these regulatory networks to basically see how genetic differences that are scattered in the genome are in fact converging into a small number of common pathways, and how we can actually manipulate these pathways, of course, first at the cellular level and ultimately, in humans. So, yeah.